It's certainly my challenge to give the next 45 minutes to talk about emerging therapies, having to realize that I've been at these meetings since they started, and 20 years ago, this talk was never on the agenda. There was no emerging therapies, and it's just amazing now what is happening. All the companies that either are involved in the MPS field already, that are doing clinical trials, or have announced they're doing clinical trials, that comes up to almost 20 different companies. It's just phenomenal what is happening in terms of the sphere. And so actually I could spend a lot more time than 45 minutes, and I'll try to keep it that. Mark, if you could give me a flag about 10 minutes to go, I'll appreciate that. So I have disclosures, as you heard. I currently consult for a variety of biotech companies. I serve on advisory boards. And as you heard, I'm also a principal investigator currently for an MPS 2, phase 1/2, 2/3 clinical trial, also for a Sanfilippo phase 2b clinical trial. And I'll currently in the future, hopefully in the near future, be involved in a variety of gene therapy clinical trials. Almost all of you are aware of this, but I'll just set it the stage because you already heard a little bit. We know what the mucopolysaccharidoses are. They do to missing enzymes, resulting in accumulation of the glycosaminoglycans. I would say that this primary event is what's really the key, the accumulation of these glycosaminoglycans. After that, all sorts of other things happen that we don't really understand in terms of secondary effects. But that initial storage is really the hallmark. And once things start to get stored and you start kicking off these cascading events, other things, it's sometimes hard to stop the process. So as you'll appreciate, in the future, the earlier we intervene, we'll get much better results, as we've seen with ERT. We all appreciate in this room, as you look around, and certainly as parents, you realize the progressive nature is either ongoing disorders. They only get worse. They never get better once they start. And one of the major challenges for new therapies, developing new therapies, is the clinical variability among different disorders and even among patients with the same disorder. Some have more involvement of neurological systems, some have more heart involvement, some have more bone involvement, and these really make it very challenging to develop new therapies. So as you appreciate, there is some current treatment options for some individuals with MPSs, either hematopoietic stem cell transplantation or IV intravenous enzyme replacement therapy. And here's just sort of what the current treatment options are for some individuals. You can see that for MPS 1, there's currently transplantation for the severe MPS 1 patient under 2 years of age, so-called Hurler syndrome. Bone marrow or cord blood transplantation is the treatment of choice. We have enzyme replacement therapy, as most of you realize, for MPS 1, 2, 4A, and 6. This is to treat the physical disease. As we already heard, the enzyme does not cross the blood-brain barrier as we currently give it. But as you also appreciate in this audience, the Sanfilippo parents have asked for a long time, why not us? And clearly that's really one of the huge unmet needs. Along with a severe form of Hunter syndrome, there is no therapy for their neurologic disease, for their progressive cognitive impairment. But that is slowly coming, and hopefully in the future we will be able to say, you've heard just about the ongoing gene therapy trial for Sanfilippo A, and I'll sort of cover some of what's happening. And these are some of the challenges we all appreciate who are involved in the clinical area of trying to develop new therapies. There is no effective therapy option for some disorders I alluded to, severe form of Hunter's and all the Sanfilippos. IV ERT currently is beneficial. Unfortunately, it requires weekly infusion by and large, and it's incredibly expensive. In the US, we're very fortunate that we don't have to worry about sort of reimbursement issues, but lots of parts of the world, reimbursement is a major issue, and there are patients who are not being treated who could benefit because of reimbursement and because of the cost. Haven't talked much about bone disease as a major component in terms of the MPS disorders, and clearly bone disease is really not impacted by IV intravenous therapy and or hematopoietic stem cell transplantation. So that's clearly another significant unmet need. As you're aware, transplantation does benefit the severe form of MPS 1 in terms of their brain disease. And that's really the treatment of choice. But it's unclear why the other disorders, that is MPS 2, the severe form, and Sanfilippo A and B, have not had the same therapeutic impact on the neurologic or cognitive impact as we've seen in MPS 1. It's not really clear, and I could spend the whole hour discussing that, but that's not the basis of this talk. So now I want to just talk about some future IV enzyme replacement therapy, things that are happening, just so you know what's out there. A company in Korea called Green Cross, who developed basically an IV form of enzyme replacement therapy for MPS II. They call that product Hunterase. That's not approved in the US, and that someday may be another option for treatment for families, but they're working on trying to develop regulatory approval around the world. For MPS 7 families, are there any MPS 7 families here? I'm not aware, but haven't seen any, but for MPS 7 families, clearly there is now some options where prior there wasn't. I should say MPS 7 is one of the rarest forms of MPSs. A company called Ultragenyx, formed by Emil Kakkis, who was instrumental in developing the MPS 1 enzyme replacement therapy, has developed a product for MPS 7, recombinant beta-glucuronidase, using IV infusion. They just recently completed a phase 3 study, enrolled 12 patients receiving 4 milligrams per kilo per infusion every other week. And they've seen some, on their initial data, they reported some positive results, and they're going to go to regulatory approval. So hope in the near future we'll have approval for MPS 7 in terms of IV ERT. Another company called Alexon Pharmaceuticals is developing a product for MPS 3B for IV ERT. They report that this product crosses the blood-brain barrier for reasons that are not clear, because most IV enzymes do not. There's been a clinical trial results reported of 11 patients where they found a 26% mean reduction in CSF heparin sulfate at the highest dose studied. They're planning to do higher doses, so that's sort of an ongoing trial to give an IV infusion to try to impact the brain disease for Sanfilippo B. Another future IV trial that's in progress is a company called Armagen from the West Coast. They have produced modified recombinant enzymes, and I'll talk about that more, for both MPS 1 and MPS 2, and those clinical trials have started, and that's what I'm going to talk about in a second. But first I want to just sort of show a slide talking about new therapeutic strategies for the MPS. There's incredible numbers of different approaches to put the missing enzyme back into cells to try to prevent the accumulation of the glycosaminoglycans and thereby prevent the progression of the disease. And so this is a whole list. I'm going to go through this in the time I have. I think I'll be able to cover most of this, but I want to go through each one. I'll talk about it, talk about some of the pros and cons, and then there clearly will be a discussion between 11 and 12 where we could have time to really take and answer more specific questions. So the first one I want to talk about, the Armagen product called the blood-brain barrier penetration of proteins. So you heard that in general the blood-brain barrier prevents large molecules from going across. This is actually a very good thing because the brain is a very sensitive organ and you don't want a lot of noxious proteins affecting the brain. You already heard this is really unique to the brain and not to the rest of the body. So what they have done is proposed to treat the brain disease using novel means, this was in the past, to move proteins across the blood-brain barrier based on the initial work of Bill Partridge. And what he has done is to take the MPS enzyme and fuse it with an antibody that recognizes the insulin growth factor, the insulin receptor on the blood-brain barrier. And when you do this, it turns out the antibody binds to the insulin receptor, and this then allows, because normally insulin gets in the brain this way. Normally insulin binds to this receptor, and you can actually move insulin into the brain. But with this modified enzyme, you basically have it now, ability to cross the blood-brain barrier and get into the brain. And what they've done, and I'll just show you one data slide. So basically they've taken monkeys, they've taken both their product, which they call for MPS 2, AGT182, and put a radioactive tracer on it, and they've also taken Elaprase, the commercial preparation made by Shire, ironate 2-sulfatase, and also labeled that. And in these monkeys, they basically gave an IV infusion. And what you see on the right is there's virtually no, and the darker the color is the more radioactivity they observe in that monkey when they've actually analyzed those tissue sections. And you can see there's virtually none going across. And in this monkey, you see actually lots of enzyme crossing into the brain where you didn't see it with Elaprase. So that's really their evidence. This doesn't mean this enzyme is working there. Remember, lysosomal enzymes only work within the cell. This shows it gets into the brain fluid, but it doesn't say what it's doing. One of the challenges for their therapy is that you can't use an animal model because in this case, this antibody is unique to the human insulin receptor, and therefore studies can only be done basically in man. All the animal models we have for MPS 1 and 2 cannot be used since their product will not work at all in those animals. So it makes it a little more challenging for development. So where are they? So Armagen has recently reported that 4 adults with MPS 2 have completed the initial dosing cohort, that is 1 milligram per kilo IV weekly, and they're now looking for patients, additional adults, to enroll in the 3 milligram per kilo cohort. They initially approached the FDA to actually do children with MPS 2, that is severe form, and the FDA unfortunately said to them, no, you have to do this in adults. As we appreciate, one of the challenges, there's not that many adults who are attenuated with MPS 2, and not everybody wants to be involved in clinical trials. And so that's one of the challenges of this particular approach, since the FDA required them to do these initial studies. Once they get their initial safety data, then it's possible then to look at individuals with a severe form. Remember, the adults have no significant cognitive problems, so the benefit to them is really much less, and they still have the inherent risk. And so this is one of the FDA sort of issues. They're not willing sometimes to give families take more risks than they think they should have. But hopefully that will slowly change with time. We're also aware that in MPS 1, they've actually treated, you can see at the very bottom here, 1 patient so far in the US has been treated, and she's at this meeting with this AGT 182, their equivalent for MPS 1. They've actually recently completed an open-labeled single-dose study, dose escalation, that is they treated patients in Brazil, giving them a single dose of the product, showing that it was safe, low dose, medium, and high, and they're now in Brazil starting to enroll patients with MPS 1 in a more of a phase 1/2 safety trial to see what sort of effect they have. One of the things in the US, we see very few severe untreated MPS 1 patients because virtually almost everybody goes to transplant. In Brazil, transplantation is not as readily available, so there's much more in the way of pediatric patients with a severe form of MPS 1 who are available for clinical trials. So that's blood-brain penetration of protein, and there'll be other means to other companies are now proposing to use other receptors on the blood-brain barrier to move proteins across, and we're going to see in the future other trials, and it's unclear which one is going to be more effective, but at least we're starting to see a movement to try to get proteins across the blood-brain barrier. So another area that's really still undeveloped, but I hope actually gets more traction in the future, is something called substrate reduction therapy. So substrate refers to the glycosamine glycans, and what we'd like to do in this case is to decrease the amount of glycans made, and therefore when you store them, you have less to store. So as you appreciate, ERT, enzyme replacement therapy, is aimed at removing the accumulating materials within the lysosome by delivering an attacked enzyme. Substrate reduction therapy design is actually to decrease the amount of GAGs that come to the lysosome, therefore you don't need as much enzyme to help prevent those from accumulating. One of the unique features for substrate reduction therapy, which is much more common in some of the other lysosomal storage diseases, things like Gaucher disease, that these typically are small molecules. As you heard, small molecules can cross the blood-brain barrier from Dr. Flanagan and have the potential to really be used after oral administration to treat brain disease, where most other things we have to give IV or we have to do direct injection into the brain itself. So again, glycosamine glycans are these large molecules that we all have. They have a variety of functions, most of them which we're just starting to understand. They mostly occur extracellular. They sort of help cells talk to one another and so-called in the extracellular space. There's some glycans that are inside the cells too. And it's important, you can't get rid of them completely. You absolutely have to. So when you talk about substrate reduction, there's a place where you can reduce how much is made, but you can't reduce it to zero since these are crucial biological compounds. So substrate reduction in itself is probably not a a standalone therapy, but hopefully in the future could be used with other therapies. And that's one of the themes you'll hear in the future, that most patients probably in the future will be treated with more than one modality, maybe substrate reduction and this or that, or another compound and IV-ERT. And so I think we're going to see that depending on what the needs are and what the disorder is. It's going to get even more challenging in terms of how to manage these patients. There is no approved medication available for substrate reduction for MPSs. It's been an area that has not had a lot of research, and clearly has been. A lot of you are aware of the compound genistein. It's actually a soy isoflavone. It's reported to inhibit GAG synthesis. It's been widely used, and a lot of the Sanfilippo families, I imagine, are using it. Can you raise a hand? How many Sanfilippo patients have their children on genistein? Some. I mean, it's clearly been used by a lot. Unfortunately, there's been no evidence based on clinical trials. There's a clinical trial that's ongoing in the UK. I was hoping we'd hear results this year at the International MPS meeting in Bonn a few weeks ago. They actually said it's going to be another year before they have results, so we'll have to wait another year to see whether genistein really has a benefit or not in terms of treating, decreasing the GAGs. By and large, I think it's relatively safe, but we just don't know its long-term side effects. There's a company that BioMarin bought out in the past that was developing small molecules, and what they've tried to do with this company, and BioMarin's continuing this research, even though they won't talk about it much, so I don't have access to it anywhere near as much as I did a few years ago when I consulted for them, Zaccharon Pharmaceuticals. What they're trying to do is to develop small molecules that really prevent the production of particular types of glycosaminoglycans. glycans. I'm going to show the next slide to try to illustrate this. So what you see here is sort of a schematic of what glycosaminoglycans glycans are. They're repeating disaccharide, repeating sugar molecules. And one of the key features, for example, for MPS II is that in MPS II, one of these repeating molecules is called iuronic acid, and that iuronic acid typically can have a sulfate on it. So the MPS II enzyme, normally MPS II enzyme is called iuronate 2 sulfatase. That is, it clips this molecule right here, and then the rest of the chain can be broken out. And so if you have Hunter syndrome, that's the enzyme you're missing, iuronate sulfatase, or also called iuronate 2 sulfatase, and then that gets rid of that. So what BioMarin is developing is a molecule that you would take, and it prevents the synthesis of this O-link sulfate. It turns out that these glycosaminoglycans, glycans, you don't have to have that sulfate on the 2 position. You just need it on other places. And so in theory, you can actually have an oral medication that totally prevents you ever to have to break down this particular. And so in theory, it could be an oral pill that that's all you would take, and you would never have any issues with Hunter syndrome, in theory. There's a lot of challenge to that, but that's where they're heading, and hopefully in the future we'll hear more. But as a lot of these preclinical studies, they don't really talk at all, and I can't even get them to say much where they are, unfortunately, even though I try. So that's so-called substrate reduction therapy. So the next one I want to talk about is another fairly complicated technology, and even a lot of geneticists aren't that familiar with it. It's called stop codon read-through therapy, and you heard a little bit about this in terms of what stop codons are in terms of Dr. Flanagan's talk. And so you all heard we have to take the messenger RNA, and from the messenger RNA that the body makes, we then make a protein from that. And these proteins have to only be in a certain length, so that as the protein's being made, there's a normal stop that occurs that we all have that says make this length protein because that's all that's functional. But we know that there's mutations called these nonsense or stop codons that are premature stops that result in us making a very shortened protein, as you heard in the Duchenne talk or the presentation a little bit in terms of that. And when these happen, you basically have no functional protein, and that then causes you to have the disorder. So historically, we've known that certain antibiotics and other compounds can allow the machinery of the cell to so-called read through this premature stop. They're very inefficient, but that's the analogy. If you can read through that, it can help. And so the next is a schematic just describing that. So the upper panel shows what normally happens in translation, where we take the messenger RNA and we make a protein. You basically have 3 nucleotides at a time. You have a transfer RNA that then adds this amino acid, and you make a full-length chain. If you have a mutation that causes a premature stop, you basically make part of that, and that truncated protein or shortened protein is basically totally inactive. So what now companies are working on is to develop small molecules that allows this machinery to read through or bypass this premature termination and make some full-length protein and some non-functional protein. But as most of you are aware, heard me speak in the past, in the MPS arena, 1 to 2, maybe 3% of residual activity is all you ever need to get a normal functioning cell. So you don't need tiny amounts. You don't need 50%. You don't need 25. 10 maybe is more than excess. 1 or 2% may dramatically affect what we have. And so now there's a drug called Adalurin that's actually been studied, but there's a number of other companies that are actually going to get into this arena. So I'm just going to highlight this because the other companies are not really yet for clinical trials. So a company called PTC has developed this Adalurin, which is much more effective in this read-through process. They've done clinical trials with Duchenne and cystic fibrosis. In Europe, Adalurin is approved for the treatment of patients with these nonsense or stop codons with muscular dystrophy. And it turns out, as some of you are aware of, that in MPS I, about 70% of the MPS I patients in the US have at least one of their 2 mutations due to a stop codon, and so those potentially one drug could treat 70%. The same drug could also be used to treat other disorders with the same type of stop codon, where, as an example, enzyme replacement therapy is unique. You have to have the enzyme for that disorder, where in this case you have a medication that can be used across a variety of different disorders, but you have to have the right genetic mutation to be eligible. A phase 1/2 clinical trial for MPS I patients using Adalurin had screened patients in Europe. Because of the difficulty of finding sort of patients, they really not have really any eligible patients yet for treatment, and it's not clear where that trial is going, but certainly the potential in the future we'll see other clinical trials where stop codon read-through therapy will be used, and this may be an adjunct, again, because this may get into bone better, may get into other tissues where IV enzyme does not get. You heard a lot about gene therapy, so I'm just going to give you a highlight of what's been out there. I'm not going to really talk about that. Again, I just want to say that what gene therapy is. Again, it's a procedure with genetic material. It could be either genomic DNA, it could be coding cDNAs, we already heard, it can be RNA, where you insert it into a cell to correct a genetic error or introduce a new function. So there's a lot happening in gene therapy these days. You already heard there's different ways to do this. You can either inject the vector directly into the bloodstream, or you can remove cells, take those cells out, in culture, affect cells, and then put them back. So there's different ways to do gene therapy. In terms of animal experience, I had previously worked on many years on MPS II and MPS IIIB gene therapy. Dr. Fu, who's really here, very proud of it, she was actually a postdoctoral fellow of mine, and actually she initially got involved in my project, and then when she came here to Nationwide a number of years ago, she took the MPS IIIB, and then they worked on A and also Hunter here most recently. Most of you are not aware, not sure if Chet's in the audience. Chet in the audience? Dr. Whitley was here yesterday, but actually many, many years ago, he treated one patient, adult with MPS II, with retroviral ex vivo. He took out their white cells, transfected their white cells with this retroviral vector expressing the MPS II enzyme, put it back. They saw really no clinical effect just because of the inefficiency of transfer of that problem and stopped that. You already saw the paper. There's 4 French 3A patients who underwent direct brain injection. In Paris, there's also been 4 French MPS IIIB patients who had a similar procedure form. That procedure appears to be well tolerated, even though it's very invasive. And they have both used an AAV vector called RH10, which is a rhesus monkey 10. And one of the advantages of this vector is that there's really no immunity, since it really occurs in monkeys. It's not a virus that occurs in man. There's a human gene therapy trial for MPS VI in the planning stage, and hopefully we'll start within the next 6 months to a year in Europe using IV AAV gene therapy that potentially could really correct and get around the issue to have to weekly infections. And you already heard here the nationwide program where there's 2 patients who have been treated for MPS 3A, and they hope to do the 3B some point in the future. One of the major challenges for the gene therapy trials, even worldwide, is the availability of vector. To be able to produce the commercial, I should say clinical grade material is very time consuming and very expensive, and it's really difficult to get enough for large amounts of patients. It'd be nice to be able to make a batch of vector that treats 10 or 20 or 30 patients. Right now it's limited, and that's what's really limiting some of the trials here at Nationwide, is really the availability of the vector, and there's really a need to improve that process. A company that was based out of Jim Wilson's work out of Penn, Regenix Bioscience, is in the planning stages to treat the brain disease in MPS 1 and MPS 2. They're going to use a direct injection of AAV vector, most likely AAV9, into the spinal fluid, most likely potentially into the basal cistern, even though I've not seen the final development. And then I want to talk about another process that's something relatively new. Sangimo Bioscience has developed a platform technology to basically use gene therapy to have the liver make large amounts of enzyme. As you may know now, the people on IV enzyme therapy, each of those enzymes require a huge manufacturing plant, $50 to $100 million plant, to make those products. So it's one of the reasons they're so expensive. And so what they're using is a so-called gene editing technique, using a technique, one of the sort of 3 common gene editing procedures out there, zinc finger nuclease technology, and they're planning trials for MPS 1 and 2, hopefully starting this year, and I'm going to be involved in both of those trials more heavily in the MPS 2 trial. I just want to show you what this is, 2 slides from Sangimo. They basically are taking the MPS gene, either it's 1 or 2, and inserting it directly into the albumin gene, and then by using a liver-specific promoter, so that the protein is only made in the liver. By doing an IV injection, they'll hope these go to the liver, insert the protein into the gene, the genetic material into the locus, and then result in high levels of expression. I just want to show you work using my MPS 2 mouse model that they have done. Basically, what you see here is use this technique on the MPS 2 mouse. Here, 3 weeks after injection, you can see baseline levels are down here, and they have incredibly supernormal levels of enzyme in their liver. If you go to the far right, you see the same thing with spleen, even though it's nowhere near as high. But more importantly, in the spleen, which is a somewhat difficult tissue to treat, they actually see normalization of the GAGs in this mouse model. And you can see here in the middle, they're making huge amounts of enzyme, many-fold above normal. Be aware that you only need 3 or 4, maybe 5% above normal, not 500 or 10,000-fold above normal. So this is potentially a technology that could replace IV enzyme replacement therapy. People have asked me, will this help the brain disease? It's unclear whether these really high levels will a little bit get into the brain. We talk about the blood-brain barrier. It is not 100% impenetrable. A little bit of, for example, albumin in the CSF, we find that it's only made in the liver, so a little bit will go across. So what's unclear, will enough enzyme go across to impact even the severe patients? Time will only tell once we get to clinical trials and see how much enzyme occurs in the brain. So in terms of anti-inflammatory therapy, another area that's been very active, Leo Seminaro and her colleagues up in New York have basically treated the MPS 6 rat with a compound called pentosan polysulfate, which was remarked in remarkable abilities in terms of their mobility, grooming behavior, and skeletal disease after oral administration of the drug. This compound, as some of you know, is already approved in the US. It's called Elmaron. It's really for the treatment of interstitial cystitis given orally. They're basically in the process of trying to see if this can be used for other MPS disorders in addition to MPS 6. There's been 2 small investigator-initiated studies in Germany and Japan with MPS 1 and 3. The results are sort of, unclear. They're talking about using a sub-Q preparation, which is much more higher dose level than the oral Elmaron dose. And so hopefully time will see some more studies in terms of this anti-inflammatory therapy, which may be an adjunct to our current therapies. So last I want to spend, got 10 minutes I guess, Mark, talking about intrathecal ERT. And so what this is basically putting the missing enzyme directly into the spinal fluid. By doing that, we literally bypass the blood-brain barrier. The indications are really two. One, spinal cord compression due to very thick dura, and then clearly the central nervous system manifestation, that is the cognitive impairment that occurs with progressive storage. Here's just an example of a patient with Hurler-Shea syndrome who has a very tight spinal cord, and one of the hopes is, can you put enzyme into around the spinal fluid and decrease that? One of the challenges is a lot of this thickening of the dura is due to fibrosis or collagen way down, and all the enzyme in the world. It's hard to get rid of some of that. So those trials have been somewhat beneficial, but not as much as we would hoped in general. Patty may talk a little more about that, so I won't say any more. I just want to say that BioMarin also now is developing a treatment for MPS IIIB, taken the MPS IIIB enzyme called NAGLU and fused it with a peptide derived from the insulin growth factor 2, and now they're using this to help get it into the brain cells. We're going to do IV, but now they're actually doing direct brain injections. This trial has actually started in Europe, has not started in the US, to my knowledge. Here's just a schematic showing a lumbar puncture where you can put enzyme into the spinal fluid or any product down low, or to use direct injection into the ventricle. This next slide just shows you what an Ommaya reservoir is. This is basically like a shunt for those patients have shunts, but there's no really tubing coming off. You put it basically a catheter through the brain tissue into the ventricle, hooked up to a reservoir, and now you can either draw back fluid or infuse. And that's what BioMarin is doing for MPS IIIB in Europe. Turns out there's lots of intrathecal ERT happening in the MPS world these days. This is a list, and I really will just very briefly sort of summarize it. I could spend a couple hours talking about all this in detail if I had the time, but we won't. I just talked about the MPS IIIB direction. There's been IT-ERT, basically for spinal cord compression for MPS 1 and 2. Drugs are well tolerated. It's just difficult because of the nature of their disease. There's been, Patty may talk about, the next speaker, Dr. Dixon, IT-ERT for MPS 1 over 5. There's also intrathecal ERT given in correlation as transplant of MPS 1. People at Minnesota have seen a cognitive decline in the first year after transplant, and the hope is by giving extra enzyme they can minimize this cognitive decline. We're going to hear data from that in the next year or two. And it's both intrathecal ERT trials ongoing for MPS IIIA and I'll talk about very brief at the end, for MPS II, which I've been heavily involved in. So some of you have seen this. I'm just going to go over it again. What the current study designed for the phase 1/2 trial. We initially recruited patients 3 to 18 years of age. They have to have cognitive impairment. We're using monthly injections of enzyme using an intrathecal drug delivery device, which I'll show you in a second. There were initially 3 doses evaluated, 10, 30, and 1 milligram given monthly. All patients were continued on weekly IV enzyme, and there were 16 patients initially recruited for this study. We initially used this Smith Medical device, which really was not robust for our very quiet Hunter patients. Not so quiet. This device actually broke right here because really of a structural issue with the way it was made. It just didn't last. This device was approved in Europe for 2 populations, pain control for brain tumors and for intrathecal chemo for brain tumors. And I joke with people, 2 populations that are not wrestling with their brothers. So the current design for the phase 1/2, it could not have increased opening pressures, it could not have communicating hydrocephalus, it could not have a shunt, and we were doing a so-called a DOS, using the DOS2 for yielding a GCA, which we refer to as general conceptual ability, which is similar to an IQ, but not really an IQ. This population we initially enrolled 4 to 11 years of age. It was primarily a safety trial, it's still ongoing. Patients had wide range of cognitive abilities, and they had lots of different mutations. We have measured serial CSF glycosamine glycans, basically looking at dermatan sulfate, and you can see dramatic reductions in all 3 doses used after 1 month. Had more effects with the 10 and 30 than we did with the 1 milligram dose, and because of that, patients were actually put onto the, everybody was on to either the 10 or 30 currently in the trial. Here's just actually the GCA scores for patients before and after treatment, so what it's like. You can see the dashed line here is where they started. And you can see there's a wide range of abilities prior to starting in terms of overall functioning. But I want to just put this on. This is an arrow that would dictate what would happen if you lost 12 IQ points per year on a continuous basis, and see what happened. And you can see that there's clearly patients in this trial who have dramatically changed what their course I would have projected to be. Again, it's impossible for any one patient to know where they're going to be. But if you just take this as what we might expect, you can see there appears to be a significant a beneficial effect in terms of this intrathecal ERT, even though it's been challenging delivering the enzyme because of the device issues we've had. So overall summary in my last few minutes is that monthly intrathecal administration of ironate IT at 10 milligrams, 30 and 1 appears to be generally well tolerated. We have not seen significant safety issues with the drug itself, and that clearly is in the current phase 2 trial. Phase 2-3 trial I'll talk about in a second. The total CSF glycan concentration was reduced after each dose. The decline we saw within single dose, we saw a rapid decline. There's a similar reduction in the 10 and 30, and so therefore, and not as good a reduction in the 1. And so all patients are currently in this trial on either 10 or 30 doses. There was 2 patients who already had antibodies against ironate sulfatase prior to the introduction of the trial. We actually made their antibodies worse in this trial, so there's potential for actually not everybody having the same beneficial effect, and this clearly decreased their ability to decrease GAGs was reduced when they had significant antibodies. So it's like anything else, the antibodies issues are concerned. These antibodies occurred in patients who really had so-called nonsense mutations. They didn't make any protein whatsoever predicted. 3 of the 5 testable patients, remember, not everybody was testable because it was a safety trial, and we early on had difficulty enrolling patients, so we basically then expanded the trial to really get patients who were not really testable. But of the testable patients whose sequential cognitive assessments are available, there's very encouraging signs, as you saw from the data, in terms of stabilization of effect. This clearly warranted the phase 2-3 trial, which is ongoing. This just summarizes that current 2-3 phase trial. This trial basically is going to close in the next probably 2 months, since we're now very close to our enrollment goal. This was initially a 1-year trial, a phase 2-3, so a pivotal study, testing 10 milligrams monthly intrathecally using a Sofista device or LPs when we couldn't use that device. We enrolled patients from 3 to 16 years of age. Typically, it's patients who are older, beyond the age of 7 or 8, or don't qualify since they're too cognitively impaired. They could not have increase intracranial pressure, and they could not have shunts. Doesn't mean that the drug would not work with those, but for the clinical trial, those were excluded. And our primary outcome from this trial will be a GCA score after 12 months of treatment, comparing patients who were randomized 2 to 1. Every 2 out of 3 got study drug. 1 got no treatment for the first year, and then those patients got treatment. And so hopefully in another 15 to 18 months from now, you'll be able to see the results of the study and see where we are in terms of what's happening. I'll be certainly happy to talk about this trial and sort of implications for future other patients treated once we have the discussion at 11. So thank you for your attention.